Thank you. Well, first I would like to thank the organizers for giving me the opportunity to present my work. My name is Karina. I'm a postdoc in Field Animal Lab at Mass Medical School. And I'm using high-throughput biochemistry to better characterize microRNA-binding rules in vitro with an ultimate goal of better understanding microRNA function in vivo. And today I will talk about our computational strategy, which provides a high-throughput rule to assess thermodynamic and kinetic properties of microRNAs, but also of other RNA-binding proteins. So microRNAs are short-non-coding RNAs loaded into RBNS proteins, which guide them to partially complementary binding sites. And once RNAs are bound, they serve as a scaffold and recruit secondary silencing factors, which trigger mRNA degradation or inhibit translation. A large portion of microRNA knockout mutants display an abnormal phenotype, underscoring the importance of microRNAs in tissue development and homeostasis. However, we currently do not know cellular targets that most microRNAs repress, and therefore we do not know how most microRNAs contribute to establishing specific developmental programs. We do know that binding specificity is conferred by a short sequence within microRNA guide, and therefore a typical microRNA can bind dozens to hundreds of cellular transcripts, and a specific transcript can be concomitantly targeted by several microRNAs. Current evidence suggests that microRNA-mediated repression is a linear function of the occupancy of microRNA binding sites. And in principle, the occupancy itself is a linear function of binding affinity between the RNA binding protein for its regulatory binding sites. And this binding affinity is expressed as equilibrium dissociation constant Kd. And so higher binding affinity leads to higher occupancy and therefore to higher repression. However, the sites with the same Kd may result from different kinetic behaviors. For example, there are some microRNA binding sites which are bound very stably as measured by slow dissociation rate constant Ko. But these sites are also found by microRNAs very slowly. Another type of site presented here are found by microRNAs much more quicker, but they also dissociate quicker. And because Kd is defined as the ratio between K-off over K-on, these sites have the same binding affinity, but they, in the first case, the interaction is stable enough to recruit secondary silencing factors and result in repression, while in the second case, dissociation is too quick, and this binding does not result in silencing. So this is why it is important to gather both thermodynamic and kinetic information to build predictive quantitative models of regulatory networks of microRNAs, but also of other RNA binding proteins. In particular, I aim to combine in vitro and in vivo data to establish integrated quantitative approaches to study microRNA function in vivo. We do know that microRNA sequence strongly influences target binding, and therefore we need to obtain diverse binding data sets in order to establish predictive models of microRNA-mediated silencing. And RNA Bind-n-Seq, which was very quickly mentioned earlier this morning, is an in vitro method which is capable of exactly providing this binding data in high-throughput manner. So RNA Bind-n-Seq was initially developed in David Siegel's lab, who studied binding of zinc finger proteins, and later adopted to RNA in Christopher Birch's lab. So this approach uses synthetic RNA oligos which have central randomized region, which is flanked by constant primer binding regions, which are required for linear sequencing. In addition, we also use DNA blocking oligos, which are complementary to the flanking to the flanking regions, because this makes only the central region single-stranded and accessible for binding, which limits experimental biases. So we incubate a large pool of these distinct RNA targets with the RNA binding protein, separate bound RNA from the unbound, and sequence the bound RNA using regular EUNA protocols. And so thermodynamic and kinetic studies both use the same framework. So we use RNA concentration constant in both studies, but in thermodynamic studies, concentration of microRNAs is variable in different binding reactions, and we measure binding at equilibrium. In kinetic studies, we keep microRNA concentration constant, and we measure binding at different variable time points. And our main contribution to improving RNA Bind-n-Seq consists in developing an analytical method which robustly de novo identifies binding sites, production binding sites, and also measures KD values as well as K1 and K-off for all binding sites simultaneously from the same datasets. So in our strategy, we consider simplified version of binding reaction between microRNA-loaded argonauts proteins and sites of interest. And we use biochemical equations to predict recovered concentration of site I in every binding reaction chain. And so, of course, this concentration, the formula of this concentration contains all the unknown parameters that we actually aim to estimate. So fortunately, on the other hand, we also have the data from immunosequencing that we can use to directly quantify this recovered concentration of every site I in every binding reaction chain. And then we run iterative search for parameters of interest in order to maximize the likelihood between the model and the data. So we define the likelihood function for sequencing reads given certain values of parameters that we are looking for. And we define the cost function as the opposite of this likelihood. So we develop the cost function into this expression, and you note that intuitively, cost function maps the data in blue into the model in yellow. So it provides one single number that we seek to minimize. And to facilitate optimization, we supply analytically computed gradients. And everything is implemented at Python and runs pretty fast. So just to show you how it looks like, the predicted concentration of recovered site I is a combination, is a pretty simple formula which has the KD values that you are looking for, as well as the background non-specific binding, as well as the free concentration of microRNA-loaded complexes. So first, we wanted to test our method on a well-controlled simulated dataset that we constructed using 4 binding sites for which we define ground truth KD values ranging from 10 to 1000 picomolar. And we set the remaining parameters to values that we typically obtain when we run low-throughput biochemical experiments. So by using these ground truth parameters, and introducing some experimental noise, we obtain these recovered sites. So then we run our maximum likelihood estimation and measure KD values in the simulated dataset that we, of course, compare with the ground truth. So our cost function converges very rapidly, and the estimated KD values are almost identical to the ground truth. So then we asked whether our estimation is robust. So we performed experimental RNA Bind-n-Seq by using very well-studied FLT7A microRNA and mouse AGO protein. And we performed 6 binding reactions. Then we constructed 6 sub-datasets in which we removed one binding reaction at a time computationally and used these sub-datasets to estimate PT. And so the pairwise Pearson correlation coefficient were extremely high, and the KD values obtained in one or the other FLIG-1 data sets were almost identical. And so finally, we wanted to compare our results with published literature. And for this, we calculated KD values for the 7 canonical sites, which are very well studied in the literature. And we observed that our results agreed very well qualitatively with the data from David Bartel's lab, who measured relative binding affinities for these sites. And we also found some other reports which performed low-throughput experiments, and our results agreed also well with those. However, you do notice some differences between RNA Bind-n-Seq values and low-throughput results. And this actually comes from technical differences between the methods. So the low-throughput methods study unique RNA sequences, which are typically a little bit longer than 20 nucleotides, while RNA Bind-n-Seq is limited to shorter motifs. And this is because reads with longer motifs are very sparse in sequencing data with coverage, which is financially acceptable. So while these methods measure unique RNA sequences, RNA Bind-n-Seq measures mean context. And we do know that flanking context affects K1 in vitro and thereby KD. And so we considered 256 sequences based on D-nucleotide composition at 5' and 3' end, flanking the site of interest. And now we observe more than 10-fold difference in values. And of course, these 250 sequences contain the unique dinucleotide context used in these studies as well. And now you can see that these values are much more closer to low-throughput results. So encouraged by these results, we decided to extend our analytical strategy to kinetic studies. But now you notice that the model concentration of the site is much more complex. And it's actually a linear combination of exponential functions, which will definitely make the estimation more fragile. So first, we tested our estimation on simulated dataset, and sure enough, we did not recover the absolute truth. So our cost function converged rapidly, but was very rapidly trapped at local minimum instead of reaching the global minimum, which is defined by the ground truth. However, RNA Bind-n-Seq preserved the relative differences between K-on and K-off, and therefore we believe that our method is still useful for the field. And when studied on experimental data sets and compared to the literature, again, we obtain the same hierarchy of fast and slow binding sites by RNA Bind-n-Seq as by low-throughput methods published in the literature. RNA Bind-n-Seq, however, does not offer the same dynamic range, and this is again because of the technical differences of the two methods. So together, RNA Bind-n-Seq de novo identifies productive binding sites, which I didn't have time to talk today about. But combined to our analytical strategy, it can measure also absolute binding affinities as well as relative kinetic parameters. And therefore, we think that RNA Bind-n-Seq will yield data for quantitative models of regulatory networks of microRNAs, but also other RNA proteins. So we benchmarked this strategy using our favorite protein ABO2 loaded with different microRNAs. But since then, this approach was successfully used using fly-1 as well as mouse MeWe, Kiwi, and sponge Kiwi proteins, as well as human PNUD1. And more generally, this approach is surely applicable to RNA and DNA binding proteins to measure their binding specificity for nucleic acids. And I will finish by thanking Philip Zamer, who is an amazing mentor and definitely helped us to become better scientists. And I also would like to thank Well, who helped me a lot with very numerous library preparations. And of course, all the Samuel Lab for their for their continued support and very fruitful discussions. So I was supported by Health Resources in Action, and the project itself was supported by HMI and NIH. And last but not least, I will share with you that I'm actually starting my own group in January next year. So if you guys are interested in microRNAs as well as high-throughput biochemistry and single molecule biophysics, please reach out. Thank you so much for your attention. And I didn't say that actually it will be here in Lyon, so if you love the city, please reach out. It's just perfect time, right?
So questions? Hi, great talk. So I'm interested in the differences in the on rates. So can you tell us what dictates the differences in the on rates? You're starting with the same concentrations. So why are there differences in the on rates?
Well, this is exactly why we love Argonaut proteins. So Argonauts actually reshape their microRNA guide. So typically, Argonaut makes microRNA guide in favor of the 5' end of the microRNA guide in a favorable conformation, which is in the stacked helix accessible in the solvent. And so these Argonauts actually prepay entropic penalty so that the initial hybridization rate is extremely fast with the target. But this does it only for the 5' end, not for the 3' end. So the guys which pair through the 5' end, they have the diffusion limited on rate. However, those which pair with 3' end have regular on rate for nucleic acids, which is 10⁶ per mole per second, versus diffusion limited rate in human cells is rather 10⁸. So that's where the difference comes from.
So you mentioned that you can apply your method also to RNA-binding proteins. You know, with RNA-binding proteins, I'm sure it's also true if microRNA structure matters.
Right.
Is there a way to extend your method to include RNA structure?
So we initially developed this method without taking into account the secondary structure at all. But obviously, when I will be doing these experiments in cells, secondary structure will be one of the parameters. So we cannot directly run RNA Bind-n-Seq taking into account the secondary structure just because the microRNAs bind through 8 nucleotide or 6 to 8 nucleotide binding sites. So because RNA Bind-n-Seq is limited to 12 nucleotide motifs so far, it does not leave a lot of space to consider the secondary structures. However, for RNA binding proteins, which bind through shorter motifs like 5 mers or 6 mers, then of course we can take this into account.